reaction. So by the end of the lecture, the student should be able, the first one, to define enzyme and related terms such as active site, apoenzyme, holoenzyme, prosthetic group, and enzyme specificity. Okay, explain the energy of activation, describe the structure of enzyme, know the mechanism of enzyme, which is what is the enzyme inhibition, and the last one, explain the classification of enzyme. Okay, so the importance of enzyme, which is important role in metabolism, diagnosis, and therapeutic. Okay, diagnosis maksudnya you diagnose, you kaji apa penyakit dia. Therapeutic apa? Untuk, you pernah dengar tak therapeutic ni apa? This word. What is therapeutic?
Something that calm the nerve.
No, not harm. Therapeutic ni dia prevention. Okay, prevention to disease. Okay, so all biochemical reaction are enzyme catalysts in the living organism. Okay, the level of enzyme in blood are diagnostic important, which is a good indicator in disease such as myocardial infarction. Enzyme can be used therapeutically, such as in digestive enzyme. Okay, so the first one, the definition of enzyme are known as biological catalysts, are important that increase the rate of reaction. Okay, dia meningkatkan kadar tindak balas by lowering the energy of activation. Okay, dengan cara menurunkan tenaga pengaktifan. Okay, dia naikkan kadar tindak balas, okay, protein ni dengan menurunkan energy activation. So they catalyze nearly all the chemical reaction taking place in the cell of the body. So not alter or consume during reaction, and they are reusable, which is you boleh guna sampai bila-bila. Okay, so they have the first one, which is active site. Okay, so which one active site? This one or this one? Kelabu ke hijau? Grey or green?
Grey.
Okay, grey. So ni tapak pengaktifan. Okay, enzyme molecule contain a special pocket or cleft called the active site. So dekat atas ni ialah substrate. Okay, so the substrate entering active site of enzyme. So they have a complete enzyme substrate complete, which is the enzyme change shape slightly as substrate bind. Okay, and then after the reaction, they are living active site of enzyme, which is they have a two product. Okay, substrate tadi ada dua, ada satu. Tapi bila reaction tindak balas ni dah berakhir, dia menjadikan dua produk berbeza. Okay. So they have a two model. Okay, log and key. And the other one apa? Ada satu lagi model. Ingat tak? Log and key and? Ingat tak? Ada log and key. Dia akan substrate ni sama dengan active site. Okay. Ada satu lagi model. Substrate ni akan dia tak sama dengan active site. Okay, apa yang satu lagi model? Okay, anyone?
Induced feed.
Okay, induced feed. Bagus. Okay, sekejap. Okay, sekejap. Alright, so the log and key model, the first one, which is explain enzyme specificity. This explain the loss of activity when enzyme denature. Okay, so the key as substrate and the log as enzyme. Okay, so they are correct feed and will react. Bila substrate dan enzim bergabung, dia akan menjadikan ada reaction ataupun tindak balas. But when they are in correct substrate, there are no reaction or no enzyme reaction. Okay. The active site has a rigid shape. Okay. Only substrate with the matching shape can fit, and the substrate is a key that fit the log of the active site. Okay, so the second one is a apoenzyme and holoenzyme. So the apoenzyme is a protein portion which is inactive, and holoenzyme is a whole enzyme which is active. So the enzyme without its non-protein moiety is termed apoenzyme and it is inactive, and holoenzyme is an active enzyme with its non-protein component. Okay, so macam mana kita nak menjadikan apoenzyme ni aktif bila ada cofactor which is non-protein portion as activator and they have a coenzyme. Okay, barulah akan menjadikan apoenzyme inactive tadi menjadikan dia aktif which is holoenzyme. Okay, clear? Boleh? Apoenzyme, non-protein moiety and inactive. Yang ni tak aktif. And then nak menjadikan dia aktif, okay, dia kena ada coenzyme and cofactor untuk aktifkan menjadikan holoenzyme which is also non-protein component but it is active. Okay, boleh clear? Boleh? Faham perbezaan apo dan holoenzyme?
Faham, madam.
Faham, madam.
Okay, so what is the important term to understand biochemical nature and activity of enzyme? So, tadi dekat sini ada cofactor. Cofactor is a non-protein chemical compound that is bound, okay, either tightly or loosely to an enzyme is required for catalysis. Okay, they have two types of cofactor which is coenzyme and prosthetic groups. Okay, so the type of cofactor, the first one coenzyme, which is non-protein component loosely bound to apoenzyme by non-covalent bond, for example, vitamin or compound derived from vitamin, and the other cofactor is a prosthetic group, which is non-protein component tightly bound. Yang ni ikatan yang kuat lah to the apoenzyme by covalent bond is called a prosthetic groups. Maksudnya cofactor ni dia ada dua. Okay, coenzyme loosely bound and prosthetic group this one tightly bound. Okay, boleh? Boleh? Clear?
Boleh, madam. Boleh.
Kalau tak faham ataupun tak clear, you just raise your hand. Okay, so the enzyme is specificity. This sangat specific, which is enzyme have varying degrees of specificity for substrate, which is may recognize and catalyze a single substrate, okay, a group of similar substrate and particular type of bond. Okay, so this is the enzyme specificity, which is for the absolute group. Okay, absolute type, there are reaction type is a catalyst one type of reaction for a single substrate. Okay, for example, urease catalyst only the hydrolysis of urea. Okay, this is specific in a absolute type. Okay, dia akan bertindak bila ada single substrate saja. Urease enzyme untuk urea hydrolysis. Okay, the second type is a group catalyst one type of reaction for similar substrate, which is hexokinase adds a phosphate group to hexoses. Okay, the third group, which is the linkage catalyst one type of reaction for a specific type of bond. Okay, so for example, chymotrypsin catalyst the hydrolysis of peptide bonds. Alright, okay. And the other important term, which is the activation energy or energy of activation. Okay, so all chemical reaction require some amount of energy to get them started. It is first pushed to start the reaction. Okay, so this called activation energy. Apa yang menyebabkan tindak balas tu bermula? Okay, so the mechanism of action of enzyme, which is increase reaction rate by decrease the activation energy. So they have enzyme substrate interaction, formation of enzyme substrate complex by log and key model, and also the induced feed model. Okay, so this one, okay, without enzyme, okay, without enzyme, okay, this man want to pull the ball, okay, which is activation energy without enzyme, okay, which is lactose here, and then glucose. The product is a glucose and galactose. So the need energy released from splitting of lactose. Yang ni yang digunakan. Okay. And the second one with enzyme, okay, they have a lactase, okay, which is the activation energy lagi rendah. Kalau sini dia perlukan tenaga yang banyak. This one ini untuk activation energy. Okay, so the need energy release, okay, sama. Tapi activation energy berkurang. Boleh faham? Comparison without enzyme. Okay, starting point dia tinggi ataupun memerlukan activation energy yang tinggi. Tapi with enzyme, okay, activation energy adalah lebih rendah. Boleh faham? Clear?
Clear, madam.
Okay, so this is what happened with and without enzyme. Okay, so starting dia samalah. Okay, and then the cause of reaction without enzyme, dia memerlukan, okay, without enzyme, this is the energy used, and this one with enzyme is lower. Okay, cause of reaction with enzyme, okay, the energy is unaffected by enzyme. Okay, so dekat sini sama, tapi activation energy berbeza. Okay, so ni yang tadi log and key, which is in the log and key model, enzyme action has a rigid shape. Only substrate with matching shape can fit. The substrate is a key that fit the log of the active site. This is an older model, but however does not work for all enzyme. Sebab dia spesifik, so dia tak bertindak balas untuk semua enzyme. Okay, so this one as active site and this one as substrate. So this is the complex enzyme substrate. Okay, the second model is a induced feed model, which is this site. The active site is flexible and not rigid. Okay, yang ni bentuk dia boleh berubah mengikut substrate. Okay, you tengok active site dia macam ni, tapi substrate dia bentuk diamond. Okay, the shape of the enzyme, active site and substrate adjust to maximize the feed, which improve the catalyst. There is greater range of substrate specificity. Okay, this model is more consistent with a wider range of enzyme. Okay, so this is the enzyme substrate complex. Okay, boleh faham Kita ada dua model, log and key and also the induced feed. Okay, for the induced feed, active site not rigid, flexible. But for log and key, there are rigid active site. Okay, boleh compare? Boleh
Boleh,
Clear?
madam.
Boleh, madam.
I think you pun dah tahu sebenarnya benda ni. So just revise apa yang you dah tahulah. Okay, so they have enzyme substrate complex, which is the first step to combine enzyme and substrate combine to form a complex. Okay, so they have an enzyme here. Okay, and then they have a substrate. Okay, and then this one is a complex one. Okay, in a second step, an enzyme product complex is formed. Okay, and then they have a transition state and this is the product, two product. Okay, substrate tadi kita ada satu, tapi bila ada transition state, dia ada yang berlaku di sini reaction and then they produce a two product. Okay, the enzyme and product separated. Okay, okay, and then the enzyme is ready for another substrate. So this one dia menjadikan dia continue je, reusable. Dia boleh guna sampai enzyme tu ada, sampai bila-bila lah. Okay. Okay, and then they have a affect enzyme activity and they have a 3 factor. Okay, what is the factor? Apa yang you tahu? Apa yang menyebabkan apa yang boleh menyebabkan aktiviti enzyme berubah ataupun efektif? Anyone?
Temperature.
Very good. Temperature lagi? Cuba. You dah tahu ni. Temperature lagi? Heat lagi?
Adrenaline.
Adrenaline. Okay lagi? Cuba dulu.
pH value.
pH lagi? Okay, tu je jawapan. Okay, so kita tengok mana yang betul. Okay, so they have 3 factors. Okay, environmental condition. Okay, betul lah apa yang you cakap tadi. Okay, and then the cofactor and coenzyme. And the other one is enzyme inhibitor. So yang the first one temperature dekat sini. The first factor. Okay, so environmental condition. Extreme temperature are the most dangerous. Okay, high temperature may denature or unfold the enzyme. Dia boleh menyebabkan enzyme tu akan denature. Okay. Okay, the second one is a pH, which is most like 6 to 8 pH near to neutral. And the other one is substrate concentration. Okay. The other factor is a cofactor and coenzyme, which is inorganic substance such as the zinc and iron and vitamin are sometimes need for proper enzymatic activity, which is iron must be present in the quaternary structure, hemoglobin in order for it to pick up the oxygen. Okay, still remember quaternary structure on the protein? Ingat lagi tak? Ingat lagi?
Ingat.
Okay. Okay, so this one for the environmental factor, okay, we go one by one. Optimum temperature, which is the temperature at which enzymatic reaction occur faster. Okay, so the optimum is this one on the human body temperature, which is 37. Okay, and pH also affect the rate of enzyme, which is optimum is around 7 here. Okay, human amylase ataupun amylase ni apa? Daripada mana? Amylase kat mana? Ileo. Okay. Okay, saliva. However, some prefer acidic or basic condition. Okay, so ada yang pepsin more acidic. Okay, trypsin basic condition human amylase, and then ada yang memilih untuk pH rendah dan ada yang memilih alkaline, okay, trypsin. Right, so depends on the enzyme. Okay, so the substrate concentration and reaction, the rate of reaction increase as substrate concentration increase, which is at constant enzyme concentration. So the maximum activity occurs when the enzyme is saturated, when all enzymes are binding substrate. Okay, so you tengok dekat sini. Enzyme is saturated, far more substrate that it can deal with. So the maximum activity, all of the enzyme is combined with substrate. So this one, dia akan meningkatkan activity enzyme. And then bila satu tahap dia dah saturated, dia akan constant saja. Okay, tapi awal dia akan starting lah. And then bila dah maximum velocity, dia akan constant sahaja. Okay. And the other factor is a enzyme inhibitor. This one. Okay, kita tengok balik. This one. So, we go to the third one. 3 factors. Okay, the third factors. Okay, so there are inhibition. Okay, the first one, reversible competitive inhibition. They have a competitive inhibitor, which is they has a structure like a substrate. Okay, kalau you tengok this one, structure active site dia sama je dengan substrate. Okay, compete with the substrate for the active site. Okay, tengok this one sama. Okay, has its effect reversed by increased substrate concentration. Okay, so dia menyebabkan compete with dia nak compete active site dekat sini, and then the effect reverse by increase. Untuk meningkatkan this one, you kena tambahkan substrate concentration, and then you kena tambahkan this one lah, satu lagi substrate, which is untuk compete kan active site, and then you akan dapatlah produk baru. Okay, so kalau this one, yang ni tak berjaya lah, yang kompetitif ni. Tapi dengan cara you kena tingkatkan, okay, increase substrate concentration. This one substrate concentration ni kena naikkan. Okay, the second one, non-competitive inhibitor or inhibition, has a structure different than the substrate. Okay, kalau you tengok this one, active site and substrate, dia berbeza. Okay, substrate dekat sini macam ni. Okay, substrate kita yang original this one. Distort the shape of the enzyme, which alter the shape of the active site. And to prevent the binding of the substrate, it cannot have its effect reversed by adding more substrate. Okay, kalau you tambah lagi substrate concentration kat sini, dia tak boleh juga untuk bagi reverse effect. Okay, so yang berjaya this one lah. Sebab kat sini dia tak boleh mengubah dia punya active site. Okay. Alright. Okay. Setakat ni boleh faham? So kita ada dua inhibition. Okay. Reversible, competitive, and then non-competitive inhibition. Yang ni untuk effect of enzyme activity. Okay. So any question? Boleh faham? Boleh faham
Boleh, madam.
Okay, so this one we proceed to the naming enzyme. Okay, the enzyme of an enzyme and the name of enzyme is in many cases and is ASE. For example, sucrase catalyze the hydrolysis of sucrose. Okay, the name describe the function of the enzyme. For example, oxidase catalyst oxidation reaction. Sometimes common names are used particularly for the digestion enzymes such as the pepsin and trypsin. Okay, pepsin dan trypsin pun adalah enzyme. Okay, some names describe both the substrate and the function. For example, alcohol dehydrogenase oxides the ethanol. Okay, so they have a classification into the 6 function classes, which is the EC number classification, which is on the basis of the types of reaction that they catalyze. Okay, so they have a 6, EC1, oxidoreductases, okay, EC2, transferase, EC3, hydrolase, okay, EC4, lysase, EC5, isomerase, and EC6, ligase. Okay. Okay, so numbering dia macam ni. Okay, and then they have a principle of international classification, which is each enzyme has classification number consists of 4 digits. Okay, example EC 2711 is a hexokinase. Okay. So macam mana dia penerangan untuk this one, explanation of for 2711. 2 is a component indicate the first one is a class which is transferase. Okay. 7 is subclass transfer of phosphate. 1 is a subclass. Okay, bawah lagi, maksud kelas ke atas, subclass di bawah kelas, and then subclass bawah di bawah subclass, which is alcohol is phosphate acceptor, and one specific name which is ATP dehexose 6 phosphotransferase, which is hexokinase. Okay, this one you tak perlu hafal lah. Cuma you kena tahulah. Okay, kita ada enam group, and then dia ada 4, consists of 4 digits macam ni. Okay, this one just for your knowledge only. Okay, so this one which is hexokinase catalyst. Okay, dekat sini. Okay, glucose and ATP energy here to produce glucose 6p ADP. Okay, this one pun you tak perlu, you just tengok saja untuk knowledge, tapi tak perlu kenapa dia jadi macam ni, tak perlu. Okay, so this one saya nak tunjuk just the reaction on the enzyme oxidoreductases, which is the general reaction is a oxidation. Okay, oxidation and reduction reaction. So they have a typical subclass which is oxidase, reductase, and dehydrogenases. So the function as oxidation, reduction, and remove 2 hydrogen to form a double bond. Okay, the second one, okay, the second class is transferase, which is transfer of functional groups. Okay, they have a transaminase and kinase, which is transfer this one amino acid amino groups. Functional group here is amino groups. Okay, transfer phosphate group kepada phosphate group. Okay, relax dulu. Tengok dulu. Okay, the third class is the hydrolysis, which is hydrolysis reaction. Okay, typical subclass here is peptidase, lipase, and amylase, which is the hydrolase peptide bonds, okay, hydrolase ester bonds in lipid, and hydrolase 1,4-glycosidic bond in the amylose. Okay, the fourth class is the lysis, which is addition of a group to a double bond or remove a group from a double bond without hydrolysis or oxidation. So the typical subclass are decarboxylase, dehydrase, and deaminase, which is the first one to remove carbon dioxide. Okay, the second one to remove water, and this one amino groups. Okay, the fifth group or class is the isomerase, rearrange of atom to form a isomer. Okay, which is to convert cis and trans isomerase, converts the D and L isomers, which is epimerase. And the last class is the ligase, bonding of molecule using ATP energy, which is synthesis, carboxylase, add carbon dioxide, synthesis, which is combine the molecule. Okay, so